Good afternoon to all of you. It's my pleasure to introduce today's speaker. We have our ex-student, Dr. Somanagouda B. Patil, SB Patil. Because his name is difficult, we just say SB Patil. And he worked here at ICRISAT for 2 years here, and he was my first agronomy student I ever guided. So I was co-chair of his guide. We are very happy to have with us Dr. Chidanand Mansoor. He was his supervisor, and he is now Dean, College of Horticulture in Bagalkot. So it's our pleasure to welcome you. Patil worked here on evaluation of some tall chickpea breeding lines suitable for mechanical harvesting. This is the one area which is now of prime importance, is a priority area for chickpea, developing chickpea varieties suitable for mechanical harvesting. And we were looking for someone who can look at the agronomic aspects, because we realized that in new plant type you will have to adjust agronomy, you have to adjust seed rate, and you have to also see the suitability for mechanical harvesting. So right time, we found Patil, and he did excellent work here, and I must say that he was really it was good to have him. He was a very sincere and hardworking student, and excellent piece of work he has done, and that helped him in getting postdoctoral fellow in ICARDA. So that's so quick. After completing that, now he has the position. He is going to join in Morocco as a post PDF with ICARDA. So this is good for us, that we were able to provide him training which was helpful to him. So with that, I invite now Patil to give us a brief about his work he has done here. Patil.
Good afternoon all. Myself S.P. Patil. Today I am going to present on my seminar topic, Agronomic Investigation on Tall Chickpea Genotypes Suitable for Mechanical Harvesting. Before that, we'll know some brief background of chickpea. Chickpea is botanically called as Cicer erantium, belongs to family Leguminaceae. It is one of the herbaceous annual, mostly grown in winter season in our tropical region, and in also summer and spring season in temperate environments. It has a taproot system. It's bear nodules of bacteria, that is rhizobium. It's help for getting it provides 70% of nitrogen to the plant by biological nitrogen fixation. The leaves are imperipinnate with serrated leaflets, arise alternately at the third node of the plant. The entire surface of the plant shoots have the small hairy structures that secrete the highly acid substances that contain malic, oxalic and citric acid, that helps for reducing the pest incident, that is Helicoverpa in chickpea. The plants having many primary, secondary, tertiary branches. It's based on plant genotypes, or you differ to genotype to genotype. In chickpea we have 5 growth habits. One is that is erect, semi-erect, semi-spreading, spreading and prostate genotype. Among these habits, erect and semi-erects are very well suited for mechanical harvesting by reducing the harvest losses. But in India, farmers, they want to increase the area of chickpea because of they generate more income of their family, because it is one of the important Indian diet food legume. So they want an easy and economical method of harvesting, because of India having very scarce labors and that not available for timely harvesting, and also that more expensive. So the farmers want mechanical harvesting, but we don't have the line that are suitable for mechanical harvesting. Then chickpea breeding has come with some lines. I have tried these lines for mechanical harvesting. These lines are relatively low yielders, but we further exploited these yield by agronomic manipulations. Based on these things, keeping these point in view, I set out some objectives. Those are study of performance of chickpea genotype under both the ecosystem, under irrigated and rainfed ecosystem, and performance of these genotype under different planting density under both the ecosystem, and the interaction effect of these planting density and these tall genotype. Then finally we tried these genotypes in mechanical harvesting and its performance and its efficiency in field condition. Finally we calculated the economics of the chickpea production system. Based on these objectives, we split 3 experiments. One is trying these tall genotype under different planting density under rainfed condition, in another experiment in irrigated conditions. In these 2 experiments, we took 5 genotypes. Among those, 4 are tall lines, that is ICV 116012 these are the tall lines, and we took a 1 JG11. It is a normal Czech variety. It's mostly grown in AP, Karnataka Pradesh, Karnataka regions, and it is took in main plot. We laid out experiment in split plot result. In subplot, we took a plant density, different plant density. Normal density is 3.3 lakh hectare. We increased 20% and 40% higher density. During my experimentation, I recorded many observations. These are the growth observations, then yield and yield attributes, and pest incidents influenced by different treatments, and these are growth indices I recorded, and some of the physiological and meteorological observations, and then physical and chemical properties of soil, and chemical properties of plant and its uptake, and finally we calculated the economics of the system. Now I am presenting some of the important parameters of my experimental result. Among different genotype, the genotype that is JG11, that is a semi-erect genotype, recorded significantly higher seed yield of around 24.5 quintals per hectare as compared to other genotypes. Among tall genotypes, that is ICCB 11604, is recorded a next best treatment for recording higher yield as compared to other tall one. With respect to planting density, the seed yield increased with increasing planting density because of more yield per unit area, but the yield per plant is reduced because of increase in planting density. With respect to interaction of genotype and planting density, planting that JG11 at higher density records significantly higher grain yield, which was followed by at 20% higher, but planting that tall genotype at higher density has got on-for result with the planting JG11 at normal density. With respect to biological yield, the tall genotype having higher biological yield because of its tall nature and the stem girth is more, so it's recorded higher biological yield as compared to JG11. But in case of planting density, biological yield is increased with increasing planting density because of more number of plants per unit area, and the interaction was found significantly higher at growing the tall genotype at a higher density. In irrigated condition also we recorded similar results, but in planting a tall genotype ICV 604 116004 recorded significantly higher yield than normal, and on-for yield, that's comparable yield with planting JG11 at 20% higher planting density, and the biological yield was as similar like rainfed conditions. A seed per plant is very important for in agronomic aspects because we assess the final yield based on seed yield per plant and also 100 seed weight. Seed yield per plant was higher in that JG11 Czech variety. It was followed by in our tall line 604. But seed yield was decreased with increase in planting density. But we got higher yield under higher density because of more number of plant accumulated in per unit area. That contributed for getting higher yield and higher density. with respect to 100 seed weight, the all tall lines having the similar 100 seed weight, but the genotype ICCV 11602 records significantly lower test weight of around 17.32 gram per 100 seeds, and but 100 seed would not differ significantly due to planting densities and their interaction with genotypes. And in irrigated condition also we got a similar trend of results. Coming to number of pods per plant, we observed more number of pods per plant in ICV 11602, which in JG11, which was in tall genotype we recorded at ICCV 11604, and lower pods per plant was recorded at ICCV 11601. With respect to planting density decreased the number of pod per plant, but it resulted in higher yield because of more number of plants per unit area. With respect to interaction, planting JG11 at normal density record higher number of pods per unit, number of pods per plant as compared to other combinations. In irrigated condition also we recorded similar results. Here also we got around 62 pods per plant in JG11 and 58 pods per plant in ICV 602. Coming to plant height is one of the important factor to assess the performance of these genotypes under mechanical harvesting. In tall lines we recorded double the plant height as compared to check variety, that is the semi-erect variety. We record around 60 or 70 centimeter of plant height in ICCV 602 variety. Further, this plant height is increased by increasing plant density. In irrigated condition, we observed around 76 centimeter of plant height in tall lines as compared to the semi-erect. We recorded only 40 centimeter of the plant height. Coming to primary branches per plant, in different stages of the crop, we record significantly higher primary branches in semi-erect genotype as compared to tall one. These primary branches are reduced with increasing planting density because of inter-plant competition for limited resource available for plant. And with respect to interaction, the interaction effect did not show any significant influence on number of primary branches. We got the similar in irrigated also. We got similar, but in irrigated condition, the tall lines having higher primary branches than the JG11. Coming to secondary branches, the tall lines recorded significantly higher secondary branches as compared to JG11. And also the second branch also reduced by increasing planting densities. We got similar result in irrigated condition also. We recorded around 17.5 number of secondary branches per plant. Coming to leaf spread, canopy spread per plant is also one of the character plant types to know the compactness of the plant. Among different tried genotype, the semi-arid genotype record higher canopy spread as compared to the tall one, because these are tall and compact genotypes, and the canopy spread is further reduced by increasing the planting density. The similar results were found under irrigation condition also. Coming to total dry matter production per plant, among different genotype, the genotype ICCV 11604 recorded significantly higher dry matter production per plant, which was followed by that ICCV 11601, because these having relatively more pods in ICV4 and the stem girth is more in these genotype, that reflected in getting higher dry matter per plant. With respect to planting density, increased in planting density, that resulted in progressive decrease in total dry matter production per plant. Coming to first pod height, it is also one of the important character for reducing the losses in mechanical harvesting. In our experimentation, we record around 40 centimeter of first pod height in tall genotype as compared to the check one around 21 centimeter. This is because of we got more harvest losses in this semi-erect genotype. Further, this plant first pod height is increased with increasing planting densities, and some of the phenophase like days to 50% flowering and days to maturity will also differ in different genotypes. JG11 having that took lesser day, fewer days for 50% of flowering and days to maturity as compared to tall genotypes. In irrigated ecosystem also we got the similar results. Coming to vegetative and reproductive growth, vegetative growth period is differed among different genotypes. Here we got around 31 days. It took JG11 took 31 days for completion of their vegetative growth, but the tall genotypes have relatively a long vegetative growth, but the reproductive growth period was similar in all the genotypes as well as check and tall genotype. Coming to light transmission ratio, the higher light absorption ratio was recorded with the spreading one, that is semi-erect genotype, around 86% of light transmission ratio. It is reflected in reduction in light transmission ratio. Further, the light transmission or light absorption was increased with increasing planting densities. With respect to interaction, growing these spreading variety or semi-erect variety at higher density record higher light transmission ratio that defected in reduction of weed density as well as weed biomass per unit per plot. Coming to wilt incident in our experimentation, we recorded at two times at 30 and 60 days after sowing. Wilt incidence was not differed significantly among different genotype, but the planting density is differed because maybe higher plant per unit area in these planting density that result in reduction in wilt incidence. Coming to pod damage in chickpea genotypes, among different genotype, the semi-erect genotype record significantly higher the pod damage percent in both the ecosystem because of these are spreading type of variety. This is spreading type of variety that self for congenial environment for movement and development of helicopter pod. Further, the pod damage was increased with increasing planting densities. Coming to economics of the system, among different treatment combination growing at semi-erect genotype, that JG11 at higher density record around 55,000 of net income as compared to other genotype, which was followed by planting this semi-erect genotype at 20% higher. But with respect to tall genotype, the planting tall genotype at higher density has recorded a comparable net income with the planting that one of the normal check variety under normal densities. Under irrigated ecosystem, here also the JG11 recorded significantly higher net income, but the tall genotype growing at higher density recorded higher yield than normal planting JG11 at normal plant density, and recorded on-for result with planting JG11 at 20% higher planting densities. Coming to experiment third, here we tried 5 genotypes under for mechanical harvesting, and this performance of mechanical harvesting was compared with harvesting of these genotype in conventionally or manually. Before or at time of harvesting, we recorded seed moisture content and straw moisture content in both the method of harvesting. We got there is no any significant difference between method of harvesting as well as genotype with respect to seed moisture and straw moisture content. But in in mechanical harvesting takes only less productive time for harvesting a fed up area as compared to conventional harvesting. Among different genotype, the genotype that is spreading took more time for harvesting in both conventional and mechanical because of its spreading nature. The overlapping of branches take some time for harvesting. That also takes more time for unproductive time for bundling and cutting. That result in higher time for harvesting. In coming to forwardal speed, the machines will harvest around 2.1 meter in a second, but a pair of women laborers, they will harvest only 0.01 meter per second. We got significantly higher clean grained in tall genotype in both the method of harvesting as compared to that semi-arid genotype. That resulted in getting higher grain purity as compared to these semi-arid genotype. The weight of seed leftover pods is more in this semi-arid genotype in both the harvesting method because the pod height is around 20 centimeter. That resulted in more leftover pods without harvesting. That resulted in higher weight under both the method of harvesting, and also the grain losses is also higher in these in this JG11 genotype as compared to tall genotypes. The weight of damaged seeds are more in JG11 as compared to tall genotypes, and also the percent of grain damage is an extent of 1% in JG11 under conventional harvesting system. That is in the mechanical harvesting, the grain damage is reduced in all the genotypes. In unthreshed pods are more in the tall genotype 11602 because the pods are very small. Small pods are in the ICCV this variety, and that resulted in higher unthreshed grain losses in both the harvesting method, both mechanical and conventional harvesting methods. Coming total harvest losses is recorded around 165 and 58 kg of that yield loss per pad in JG11, but we received only less harvest losses in other tall genotypes. In mechanical harvesting takes higher actual field capacity. It's harvest around 0.58 fads per hour. In case of conventional harvesting, only we harvest around 0.008 fad per hour. It's resulted in higher operation cost of harvesting as compared to other genotype and conventional harvesting. The JG11 recorded a lower harvest percentage as compared to tall genotypes that resulted in lesser performance of harvesting performance as compared to tall genotypes. Coming to field efficiency, the field efficiency is higher in mechanical harvesting as compared to conventional harvesting. In conventional harvesting, the lower field efficiency was recorded with JG11 because of more leftover pod in mechanical harvesting, but higher in case of the conventional harvesting. The energy consumption is almost similar in both mechanical and conventional method of harvesting, but among the genotypes, the JG11 recorded higher energy consumption for harvesting as compared to other because it takes more time for harvesting. Coming to the economic analysis of harvesting methods, for harvesting of machine harvesting, we got cost of around 1700 to 2200 in tall genotype, but in case of uh, semi-erect genotype, the cost of harvesting is around 7 because of higher monetary losses in genotype JG11. That resulted in benefits of around 1100 to 1200 over a conventional harvesting in tall genotype, but it is in negative in case of JG11. Coming to practical utility of my research. Growing chickpea under irrigation condition recorded higher seed yield and other growth parameter as compared to rainfed condition. The planting a semi-erect genotype at higher plant density of 40% and 20% record higher grain yield as well as other growth parameter. But planting this tall genotype at higher planting density of 40% recorded a comparable yield at planting that genotype 20% and higher yield than planting the semi-erect genotype at normal densities. Coming to mechanical harvesting a semi-erect genotype JG11 recorded around 20% of grain losses compared to tall genotype. Finally, planting a tall genotype ICCV 11604 at higher density under irrigated ecosystem produced higher seed yield compared to JG11 by minimizing the cost of harvesting, cost of production by reducing the harvest losses. This is one of the appropriate agronomic practices that helps to farmer to get higher productivity in chickpea. There is some further studies required to further exploit the yield of these tall lines. They need to improve further productivity of tall by nutrient management and need to conduct this type of study, a different tall genotype study in different agroclimatic zones in India, and further efforts are need to be breed tall genotype with still higher yielding potential. The sum of the photography of my experimentation. This is the sowing of experiment 1 and 2. Then sowing of experiment 3 we did by tractor, and maintenance of required plant population at 21st day. It is general view of the experiment 1. It is general view of experiment 2 under irrigated condition. It is general view of the experiment 3, and it is the procedure for leaf area measurement, and we recorded light transmission ratio and then chlorophyll content and leaf water content, and collection of soil sample for knowing the soil moisture content in soil. And this is the ICCV 11601 plant, and it is ICV 11602 and ICV 1603. Then this is the best tall line. This is our that JG11 check line, and it is a manual harvesting. First cutting and then collection of plants and bundling, then transportation to the threshing yard, then threshing by mechanical thresher. And this is photography of mechanical harvesting of chickpea and collection of seed. Finally, I would like to thank my chairman and co-chairman for providing this better opportunity to work here, and I am very thankful to chickpea breeding and all staff of ICRISAT. Thank you.
Okay. Now it's open for discussions. So you have any queries, clarification questions? Please go ahead. Everything is clear, no questions.
Thank you, Patil sir, for your nice presentation. So I'd like to know what is the minimum area on which mechanical harvesting will be economically viable.
For minimum area means, or at least we require around 10 acre. Means it's minimum. It's economical for farmers. because if you have only 1 acre, 2 acre, there is no need to go for mechanical, because the 2 to 3 laborers are required for 1 day to harvest 1 or 2 acre.
Well, thank you, Patil,
Okay.
for a nice presentation. Okay, good.